Okay, kids, it's our last video for biochemistry. The crowd goes wild, okay? We've talked about the intro to biochemistry. We've talked about the carbs. We've talked about the lipids. We've talked about the proteins. And finally, it's the nucleic acids. So what elements are involved? C, H, O, N, and now phosphorus, P. Let me write out phosphorus in case you think that's potassium. Potassium's okay. Phosphorus. Phosphorus. The purpose of nucleic acids are genetic information. Genetic info. Okay, these are going to be our DNA and RNA, and that's really the two that are out there, and that's it. The cell parts involved, obviously, the nucleus, okay, is where we find the DNA, definitely. DNA cannot leave the nucleus. The only time you see DNA out of the nucleus is in a prokaryotic cell, those cells that don't have a nucleus, like bacteria. But you can also see its co-partner, RNA, and you can see that in ribosomes. You can see that also out in the cytoplasm. You can also see RNA in the nucleus as well, too. So, but there's our three things. Okay, the nucleus, the ribosome, and cytoplasm. Nucleic acids go fast. Isn't that nice? All right. So what's the monomer of a nucleic acid? Well, that's what this whole thing, or monomer of a nucleic acid, is called a nucleotide. And that's what this whole thing is. The whole thing is one nucleotide. The whole thing is like one bead. Do I dare get the beads out again? I dare get the beads out again. One bead. Okay, that's like the whole nucleotide right there is one bead. But what's inside that nucleotide? Phosphate, sugar, and a base. Phosphate, sugar, and a base. Bum, bum, bada, bada, bum. Phosphate, sugar, and a base. Pound it. Bum, bum, bada, bada, bum. Phosphate, sugar, and a base. Phosphate, sugar, and a base. 3 parts of a nucleotide. The phosphate, that's where the phosphorus is. It has phosphorus and has some oxygen in it. Sugar. This is not your regular sugars that we've talked about, like glucose and galactose, but it does still end in ose. And look at this. It's called deoxyribose here. That's the one that's in DNA, and you'll see the difference in a moment. Guess what sugar is in RNA? I'll let you think about that. Then there's a base, and that base could be an A, it could be a T, it could be a G, a C, or even a U, okay? Now, it doesn't say or U here because this is deoxyribose. We'll see in a second why, okay? So that base, and this A, T, G, and C stands for like adenine. Not that you need to know these names. I'm fine with you just saying A, T, C, G. Thymine. But I'm going to give them to you for those of you who will lose sleep over that. Guanine and cytosine. The one that you'll find in RNA that's not listed here is called uracil. Uracil. And so we're mentioning it now. Deoxyribose is a sugar in DNA. But if this sugar was ribose, then that would be the sugar in RNA. And both DNA and RNA have the same kind of phosphate group. So basically, this nucleotide will have a phosphate. It'll have a sugar. If that sugar is DNA, it's definitely going to be a deoxyribose. It's definitely going to be DNA. And this could be an A, a T, a C, or a G. But if this sugar is ribose, then this could be an A, a T, a G, an A, a G, a C, or a U. Okay, so thymine and uracil are kind of like the ones that are out or in, depending on if it's DNA. So the next picture is going to show that difference. But let me say that this is one. This is the monomer form. So the polymer form is going to be many, right? Well, it's a little different, but very much the same. So polymer form is called, nucleotide is the monomer. The polymer form is called the nucleic acid. Okay, remember NA, DNA, RNA, and there's only 2 nucleic acids out there, DNA and RNA. Okay, so we're going to look at the difference between them. And I'm actually going to start at the bottom of this. Okay, DNA, remember we said nucleic acids are made up of nucleotides. DNA is a double strand of these nucleotides. So imagine you have two of these chains and then it twists. That's what DNA is. It's a twisted double strand of nucleotides. But RNA is only a single strand. Okay, simple as that. Okay, and so let's write that down. So we have, and you can write this instead of drawing all this, you can write it. DNA is double-stranded. RNA is single-stranded. The next thing, remember the bases? Look in DNA. It can be T, A, C, or G. T, A, C, or G. Okay? But in RNA, it has to be U, A, C, or G. Okay? No big deal there. Okay? I want you to notice something about the bases. Down in DNA, because there's a double strand, you're going to see the T always matches with an A, and the G always matches with a C. C with a G, A with a T, T with an A. Okay? Later on, when we get into like deciphering DNA and seeing like how it works, that'll be very important, that code of matching there, those complementary bases. Okay? So DNA is double-stranded, RNA is single-stranded. DNA has T, A, C, or G. RNA has U, A, C, or G. Then finally, the sugars. Remember we said deoxyribose is in DNA, ribose is in RNA. Look at the tiny little difference here. This has just an H. This has an OH. That's why they call it deoxy. It's missing an oxygen. Ribose has the oxygen. Deoxyribose doesn't. So when it comes to sugar, ribose for RNA and deoxyribose for DNA. And those are the differences between DNA and RNA. And we're done. Okay? We are all done. Nothing more needed. Okay? That's it.